I'm with Dr. Mark Ungless, who's a Royal Society University Fellow working at Imperial College, and he is researching dopamine neurons, and I have a particular interest in dopamine neurons as I was diagnosed with Parkinson's disease 5 years ago. And these neurons are the very ones that fail when one has Parkinson's disease. So I'd like to start off by asking why you're interested in dopamine neurons.
I mean, for two reasons really. One is that they're so fundamentally important for normal brain operation, and so they're involved in a number of clinical disorders. So you mentioned Parkinson's disease, but also a number of drugs target the dopamine system. For example, antipsychotic drugs are dopamine receptor antagonists, and so that suggests that dopamine neurons are hyperactive in schizophrenia, or at least some dopamine neurons maybe. And then, for example, all addictive drugs increase dopamine in the brain, and it's thought that long-term changes in the dopamine system may be involved in addiction. And so although this is a relatively small population of neurons, they seem to be really important for normal brain functioning. But I think the other reason I'm really interested in them is because they just have a really fascinating physiology and anatomy. I mean, to give you an example, for the anatomy, although in the human brain there are only about 500,000 of these neurons, which of course is relatively trivial, there are about 80 billion neurons in the human brain. It's estimated that, for example, substantia nigra dopamine neurons, each individual one may be making 1 or 2 million synaptic connections. They have incredibly complex connectivity with the rest of the brain. And that's not well understood, and that's something we're very, very interested in. And the second reason is that they have really interesting activity under normal circumstances. So they're activated by rewards, particularly unexpected rewards. So they show very quick excitations in response to unexpectedly good events, and they appear to reduce their activity in response to unexpectedly bad events. So they provide this, what's termed a prediction error signal to the brain, and that's really fascinating to me.
You've kind of explained where they hang out and what they do roughly, and I'm very interested in the reward aspect of dopamine and the way that this works. Could you just give me an example or a couple of examples of what you meant by that?
Yeah, sure. So, I mean, primary rewards are things like food, drink, sex, and then people typically talk about secondary rewards, and for humans, money is really a key one. And it seems that the dopamine system is providing a kind of signal that's related to reward as this overall idea. There aren't specific neurons that are activated by food or water. It's any type of reward, anything that falls within that category, or any stimuli that predicts that. So, for example, money, which predicts the opportunity to obtain primary rewards, that's activating the dopamine system. It's not giving you detailed information about the type of reward. It's just telling you something within that category.
Anecdotally, I know from the people that I know personally who are on dopamine agonists, pretty much every person who takes dopamine agonists experiences problems with obsessive compulsive behavior in various ways, whether that be gambling, sexual behavior, food, shopping.
So this is exactly the kind of thing that people have suggested, is that the system is being duped into thinking, for example, that there are lots of unexpected rewards happening when they aren't really. These are the cell bodies of the dopamine neurons, more medial parts of the ventral tegmental area, and then going out here is the substantia nigra. And this is the region that degenerates in Parkinson's disease.
How do you identify the different neurons and their particular behaviours? I mean, is it particularly difficult?
So this particular question is something that I've been involved in for a while, because it is quite a difficult challenge. So, for example, you want to measure electrophysiological activity from a dopamine neuron, and you put a microelectrode in that region where the dopamine neurons are. Even within that region, at least 30% of the cells there are not dopaminergic. And so the question is, how do you find a dopamine neuron? And that's technically very difficult. Looking at these slides under these fluorescence microscopes is always, I never really tire of it, I have to say. They look so beautiful.
Okay, so you're a Royal Society University Fellow. What exactly does this entail, and how useful is this being for your research, and what is it actually allowing you to do?
So the Urf Scheme is a fellowship scheme for relatively young scientists, typically when they're starting out their own sort of independent research career. For me, it was, I think, vital. I got it when I was trying to become independent and set up my own lab. And at that time, it's really difficult to, you're trying to get funding, and it's quite a struggle. I mean, I think for everyone, it's a struggle to really establish yourself. When I got that award, it was really life-changing for my research. I I think really it's the long term and the flexibility of the funding there, relatively unusual and hard to come by.